picture to go with that. In the cytoplasm, there's a receptor. The hormone does cross the cell membrane and bind the receptor in the cytoplasm. So, to be perfectly honest with you guys, there are 3 to 5 mechanisms, depending on how we count, but we have to consider only 2 to understand it, the level that it's necessary for us to understand it at. The hydrophilic mechanism, meaning the hormone binds a membrane-bound receptor, versus the hydrophobic mechanism, meaning the hormone binds a cytoplasmic receptor, because of the nature of the cell membrane. And I don't think you need to or even want to, but you can certainly go back and read chapter 3 in your A&P book if you want to, and refamiliarize yourself with the phospholipid bilayer. Again, I don't think you need to do that. So the classification part simply tells us which groups of hormones are philic versus phobic. So the catecholamines and the polypeptides, as you can see here, are hydrophilic. Now you know what a polypeptide is. It's a chain of amino acids. You could just as well call them proteins. Catecholamines, on the other hand, you may not know what those are. Catecholamines are molecules that are made from tyrosine. So if something is made from, and tyrosine is an amino acid, just like glycine, tryptophan, etc. Tyrosine is one of the 20 amino acids. So if something is made from the amino acid tyrosine, it is by definition a catecholamine. An example of something made from tyrosine is epinephrine or norepinephrine. All right, steroids. You probably remember this from 201. Steroids are hormones made from cholesterol. So if the hormone is made from cholesterol, it is by definition a steroid. And you can, again, this probably remember this from last semester. This is chapter 2 in your book. And I realize that it's hard to read the words. This is estrogen, this is testosterone. But they look pretty much the same, don't they? And they look about the same as cholesterol. Now, when you really inspect them, they're not identical by any means. But as far as all of them being made from 4 rings, 3 of the rings are hexagons, 1 of the rings is a pentagon. And you could put cortisone, hydrocortisone in that same picture, and then they all look pretty much the same. So steroids are hormones made from cholesterol. All right, let's address these mechanisms now. Got time. The hydrophilic mechanism is the one we'll look at first, and it's one of the one of the coolest things you'll ever see in your life, physiologically speaking. Give yourselves 4 sheets of paper, turned sideways, duct taped together. Give yourselves a clean sheet of paper turned sideways. Most of what I'm going to draw will fit on this board. I'll have some other little sidebars and things over there, probably. Oh, definitely. So using 2 lines to represent the cell membrane, as I've been doing this morning, because it is a phospholipid bilayer. The hydrophilic mechanism, also called the cyclic AMP system, also called the second messenger system. One more time, because that's a little messier than usual. Hydrophilic mechanism, also called the second messenger system or cyclic AMP system. All right, here's our receptor. The red thing is a G linkage protein. And that G is as in A, T, C, G, adenine, guanine, thymine, etc. That blue thing is an enzyme called adenylate cyclase. So there's a lot of stuff there, and it goes back to, you've got it in front of you, I erased it, but it goes back to the fact that the hydrophilic hormones can't cross the membrane. They can't get inside the cell. They have to have a membrane-bound receptor. All right, so hormone binds the receptor and these hormones they're all deposited in the blood. They go everywhere. They encounter cells all over the body. If the hormones are hydrophobic, they're actually going in and out of cells. If they're hydrophilic, they can't go in. And they know when they encounter a target tissue that is a tissue they're supposed to act on because there is a receptor there. The hormone binds the receptor. Now the hormone receptor complex acting through the G linkage protein activates adenylate cyclase. So prior to the hormone binding the receptor, the enzyme was already there, but it was inactive. So we could diagram this using reaction terminology as adenylate cyclase subscript IA. What does that stand for? Inactive. Lower that a little bit. So inactive adenylate cyclase becomes active adenylate cyclase. And what activated the enzyme? Come on, guys, wake up.
Catalyzing reaction.
Hormone receptor complex acting through the G protein. Now, while that's right there, let's take a minute and review something from Biology 111 or high school biology, both. When you have a reaction, call this reaction terminology. When you convert A to B, what do you call A? Substrate, is that right? What else?
Reactant.
What do you call B?
Product.
Producer.
And what's E?
Enzyme.
Nothing difficult about that, but you need to, be comfortable, familiar with those words. Now, active adenylate cyclase hydrolyzes ATP. Again, I use this board for sidebars. It's not the Biology 111 hydrolysis of ATP. You studied ATP in Biology 111. You took ADP, an inorganic phosphate, and you had a source of energy, and you put that third phosphate on ADP, making ATP. And basically, your source of energy was glucose. You might have talked about oxidation of fats, I don't know. And then, of course, the reason for doing that is when you need energy, you split or hydrolyze the ATP, generating ADP, inorganic phosphate, and the free energy is released and available to do work. Well, what we do here is a little different. If you picture this as adenosine has 3 phosphates, correct? How many phosphates do we have here? 2. So adenosine with 2 phosphates, and there's our third phosphate. Well, what we do here is a little different. We split the ATP break that bond rather than breaking that bond. So we get AMP. So what's the M stand for? Mono, tri, di, mono. The 2 inorganic phosphates that we cleaved off are still connected to each other. That happens right here. Camp, as people call it, is the second messenger. Look at the name of the system. Second messenger system. Camp system. So camp is the second messenger, comma, the intracellular messenger. Remember, the hormone can't get inside the cell. So all this stuff we've done in the past 20 minutes or whatever is just to generate an intracellular messenger. What are we talking about? Big picture? No, not hormones. That's the small picture. We're talking about chemical messengers, aren't we? Hormones are chemical messengers. Well, this chemical messenger, because it's hydrophilic, can't get in the cell. And all this stuff that has happened is just to generate a messenger inside the cell.
So if it generates cyclic AMP, where does the differentiation come from inside the cell? So like, there's different hormones that bind to the outside, but if they all generate cyclic AMP, how does the cyclic AMP
I'm
go?
going to tell you. You're going to wish that I didn't.
Okay.
Now I'm going to make it very, very simple. So it is kind of like, I always, it'll be the fourth semester in a row that I couldn't help but smile when I told this story. It's kind of like if Rachel left her lights off, and some people register their car with the campus police, and so they could look at your tag and go into the little office over there and type your tag into the computer, and your schedule would come up. And so they come here to tell you your lights are on. Well, they're very respectful, and they don't come in the door. They stand there and wait on me to see them. And when I see them, I open the door. They hand me a note. So the cop is the extracellular messenger who can't get in. He can get in, but they just don't. He gives me the note. I'm the intracellular messenger. You see how it works? I say this is the fourth semester in a row where every time I tell this story I smile, because 4 semesters ago, right now, cops showed up with the note that Rachel left her. Wasn't Rachel. But anyway, 4 semesters ago, right after telling that story, it frickin' happened. And I thought you guys staged it, except you didn't know I was going to tell that story, and you guys thought I had staged it. It was crazy. Just a coincidence, though. Okay, so now you see why I said 3 or 4 sheets of paper duct taped together.
I forgot my duct tape. Can I borrow yours?
Yeah. Duct tape does a little work.
Yeah.
Can you believe the stuff I've learned to bring to class over the years? Band-Aids, Purell. Here comes your answer. The previous 3 steps are common to most hydrophilic hormones. The remaining 12 to 15 steps are unique to each different hormone. That's kind of the answer to your question. If it's hydrophilic, this is what happens. But what happens next depends on which hydrophilic hormone it is. Now, clearly, we're not going to learn or even look at 12 to 15 steps for a bunch of different hormones. That would be a different class. That would be a class called endocrinology, which is awesome. It's a pretty fun class if you're interested in this stuff. As such, we will look at the remaining steps in a generic fashion. All right, so hormone binds the receptor, hormone receptor complex acting through the G linkage protein activates adenylate cyclase, active adenylate cyclase hydrolyzes ATP, generating the messenger that is inside the cell because the hormone could not get inside the cell. You with me? What happens from here depends on whether it's epinephrine or whatever hydrophilic hormone we were to talk about. So we've looked at this. Basic biology. What is A? Reactant or substrate. What is B?
Products.
And what's the E? The enzyme. And enzymes are biological catalysts. The other thing we need to recall and hold on to we typically have abundant substrate, whether we're talking about a protein in our blood or whether we're talking about a protein in a cell. There are many examples of inactive things in our cells or in our blood, and they're just there. They're not doing anything unless they get converted to something active. So in step 4, cyclic AMP is going to convert A to B. There's all the A in the world, abundant substrate. In step 5, B is going to convert C to D. In step 6, D is going to convert E to F. In step 7, F is going to convert G to H. Now you will see why I wanted to make sure that you could use the terms that you use with reactions. So in step 5, tell me the enzyme. B. What was B in step 4? Okay, so did I understand you to tell me that while B is the product in step 4, it's the enzyme in step 5?
Yes.
And you are correct. And is that not what you see happening all the way down? So what is F in step 6?
Product.
And does F play a role in step 7?
Yes.
It is the enzyme in step 7. All the way down. $12 or 15 steps of this. This is called a cascade. And yeah, it means exactly what you think it means. Water cascades over the wall. Or I set up a bunch of dominoes here. I know these aren't dominoes, I'm not that stupid. And I push one and they all fall down, Okay. Why do we want a cascade? Well, I will tell you. Amplification. Didn't I tell you guys this would be one of the most fascinating physiological concepts you've ever seen or heard of in your life when we started this? It's about to get fun. All right, back to our biology basic stuff we just got to hang on to. What do you know about enzymes? They're biological catalysts. I know you know that because I reminded you a minute ago. Are they consumed? Are they used up?
No.
They're not, are they? In other words, I could be an enzyme. These markers are my substrate, my A. And I'm the enzyme. I take the cap off the marker. The uncapped marker is my product. I can do that again, can't I? I'm not consumed, altered, used up, modified, changed. What do you know about enzymes? They are, quote, used, released unchanged, and recycled. If you don't know that, write it down. Enzymes are used, released unchanged, and recycled. They are not consumed. They are used, released unchanged, recycled. They are not consumed. They are used. They're biological catalysts. They catalyze a reaction. They're released unchanged. They're recycled. So how many of these markers could I uncap? As many as there are, right? So how many A's do you think cyclic AMP can convert to B? When this cyclic AMP converts this A to this B, is this cyclic AMP messed up, used, changed, modified? No. So could the one cyclic AMP convert 10 A's to 10 B's? Sure it could. And we're going to use 10 because it makes the math matter. So the one cyclic AMP converted 10 A's to 10 B's. Now, here's the amplification. What if every B acts on 10 C's? Okay, one cyclic AMP was able to convert 10 A's, so couldn't one B